We are going to see topic biotechnology and its applications. Biotechnology deals with industrial scale production of biopharmaceuticals and biological using genetically modified microbes, fungi, plants and animals. Application includes therapeutics, diagnostics, genetically modified crops for agriculture, processed food, bioremediation, waste treatment, and energy production. The main three critical research areas of biotechnology includes providing the best catalyst in the form of improved organism, usually a microbes or pure enzyme, creating optimal conditions through engineering for a catalyst to act, downstream processing technologies to purify the protein or organic compounds. Biotechnological applications in agriculture, food production can be increased by agrochemical based agriculture, organic agriculture, genetically engineered crop based agriculture. Green Revolution successfully increased the food production many folds by using better management practices and use of agrochemicals, fertilizers and pesticides. Further increase in production is not possible by using these methods. To overcome this, genetically modified crop is used. Plants, bacteria, fungi and animals whose genes have been altered by manipulation are called genetically modified organisms. GM plants have many applications. Made crops more tolerant to abiotic stresses, reduced reliance on chemical pesticides, helped to reduce post-harvest losses, increased efficiency of mineral usage by plants, enhanced nutritional value of food, example vitamin A enriched rice. Application of biotechnology in production of pest resistant plants. Pest resistant plants decreases the amount of pesticide used. Bt toxin is produced by a bacterium called Bacillus thuringiensis. Bt toxin gene has been cloned from the bacteria and been expressed in plants to provide resistance to insects without the need for insecticides, in effect created a biopesticide. Examples are Bt cotton, Bt corn, rice, tomato, potato and soybean etc. Bt cotton, Bacterium Bacillus thuringiensis produce protein that kills certain insects like lepidopterans, coleopterans and dipterans. Bacillus thuringiensis produce crystals that contain a toxic insecticidal protein. This toxic protein present in bacterium has inactive protoxins, but as soon as insect ingests the inactive form, due to alkaline pH of gut, it converted into an active form of toxin and bind to surface of midgut epithelial cells and create pores that cause cell swelling and lysis and eventually death of insect. The gene from Bacillus thuringiensis has been incorporated into several crop plants like cotton, maize, rice, etc. The toxin is coded by a gene named CRY. The protein coded by the genes CRY1AB and CRY2AB control the cotton bollworms. CRY1AB controls corn borer. Pest resistant plants. Nematodes like Meloidegyne incognitia infects the roots of tobacco plants and causes reduction in yield. The infestation of these nematodes can be prevented by the process of RNA interference. RNAi is present in all eukaryotic organisms as cellular defense by silencing of specific mRNA due to complementary dsRNA molecule that bind to and prevent translation of the mRNA. The source of complementary dsRNA may be from an infection by viruses having RNA genomes or mobile genetic elements that replicate through RNA intermediate. Nematode specific genes were introduced into the host plant using agrobacterium vectors. The parasite could not survive in a transgenic host expressing specific interfering RNA. Biotechnological applications in medicine. The rDNA technological processes have made immense impact in the area of healthcare by enabling mass production of safe and more effective therapeutic drugs. At present, about 30 recombinant therapeutics have been approved for human use the world over. In India, 12 of these are presently being marketed. Genetically engineered insulin. Adult onset diabetes can be controlled by taking insulin at regular intervals. The main source of this insulin was isolation of insulin from animals. Nowadays, insulin can be obtained from bacterium using techniques of biotechnology. Insulin was earlier extracted from pancreas of slaughtered cattle and pigs, but insulin from these sources develops allergy or other types of reactions to the foreign protein. Insulin consists of two short polypeptide chains, chain A and chain B, that are linked together by disulfide bridges. In humans, insulin is synthesized as a prohormone, which contains an extra stretch called C-peptide, which is absent in mature insulin. The main challenge for production of insulin using rDNA technique was getting insulin assembled into a mature form. An American company, Eli Lilly, in 1983 prepared two DNA sequences corresponding to A and B chain of human insulin and introduced them in plasmids of E. coli to produce insulin chain. Chain A and chain B were produced separately, extracted and combined by creating disulfide bond to form human insulin. With this, I conclude. Thank you.